Scientists erase heart disease defect in human embryos. A group of scientists in Oregon has successfully modified the genes of embryos using CRISPR, a cut-and-paste gene editing tool, in order to correct a genetic mutation known to cause a type of heart defect. The experiments, which were described today, August 2, in the journal Nature, were conducted by biologist Shaukat Mylipov and colleagues at Oregon Health and Science University in Portland. Mylipov conducted the experiments on dozens of single-celled embryos, which were discarded before they could progress very far in development, MIT Technology Review reported last week when the results were initially leaked. This is the first time that scientists in the United States have used this approach to edit the genes of embryos. Cut and replace. The CRISPR/Cas9 gene editing system is a simple cut-and-replace method for editing precise spots on the genome. CRISPRs are long stretches of DNA that are recognized by molecular scissors called Cas9. By inserting CRISPR DNA near target DNA, scientists can theoretically tell Cas9 to cut anywhere in the genome. Scientists can then swap a replacement gene sequence in the place of the SNP sequence. The replacement sequence then gets automatically incorporated into the genome by natural DNA repair mechanisms. In 2015, a group in China used CRISPR to edit several human embryos that had severe defects, though none were allowed to gestate very long before being discarded. The Chinese technique led to genetic changes in some, but not all of the cells in the embryos, and CRISPR sometimes snipped out the wrong place in the DNA. Major advance. The new results are a major advance compared with earlier efforts. In the new experiments, scientists eliminated the off-target effects of CRISPR/Cas9. The team used dozens of embryos that were created for in vitro fertilization, IVF, using the sperm of men who had a severe genetic defect. The sperm contained a single copy of the gene MYBPC3, which confers a risk of sudden death and heart failure due to thickening of the heart muscle known as hypertrophic cardiomyopathy. In the new experiment, the team used CRISPR/Cas9 to snip DNA at the location of the defective MYBPC3 gene in the fertilized eggs. Most of the embryos naturally repaired the break in the DNA by substituting the normal version of the gene, which originated in the egg. About 2/3 of the embryos did not contain the mutated version of the gene, and the team also eliminated the risk that some, but not all, of the cells in the embryos contain the edited genes. In general, editing the germline, meaning sperm, eggs or embryos, has been controversial, because it means permanently changing the DNA that is passed on from one generation to the next. Some scientists have called for a ban on germline editing, saying the approach is incredibly risky and ethically dubious. However, a National Academy of Sciences report published earlier this year suggested that embryo editing could be ethical in the case of severe genetic diseases, assuming the risks could be mitigated.